axon terminal, I just kind of drew it kind of ballooned out here, right? Everything we've talked about before was coming down this way, right? This is the end of the road for the axon, right? We call these axon terminals. Sometimes they're called synaptic end bulbs. Sometimes they're called terminal buttons or whatever, right? But this is the presynaptic neuron, right? Here is the synapse. There would be out here, over here somewhere, right, a postsynaptic
cleft
cell. No.
Cell.
This cleft is the space. This is the synaptic cleft or gap here. Okay. Right? That makes sense? That's what we're looking at. We know that what we have coming down through here, right, is I'll just draw it like this. If I draw it like that, you know what it is, right? What is that? Action potential. It's an action potential spike, right? And it's traveling down here. It's a property of the membrane. And as long as there are voltage-gated sodium and potassium channels, which there are, this is going to depolarize this whole membrane. And then we're done, right, when this membrane's depolarized. We know what this should trigger is the release of neurotransmitters into the synaptic gap, which bind to receptors on the postsynaptic cell, and then give me another postsynaptic potential here. Or if this is a gland, the gland secretes something. But if it's a neuron or a muscle cell, it generates another electrical signal. Now, where these two cells communicate is at a synapse, right? That's what we're talking about. And we mentioned the other day that there are two types of synapses depending upon how the information passes from cell to cell. If the information passes just directly as an electrical signal, it does it through gap junctions. That's not what that is. Gap junctions are where the two cells are physically held together. And if the two membranes are touching, if this one's depolarized, this one gets depolarized. Very rapid, faithful spread. What that doesn't allow for, it doesn't allow for any plasticity, right? It doesn't allow me to break and reform connections like this one does. So most of the synapses in the body, we use neurotransmitters that pass across the synaptic gap. Okay? Now, neurotransmitters are going to work differently on the postsynaptic cell. Let me introduce some terms for you here. So, this is the postsynaptic cell. We are going to have a postsynaptic response. And we're going to get a postsynaptic response because in this postsynaptic cell, there is going to be a receptor here, right, that is going to interact with a neurotransmitter out here, right? And when the neurotransmitter binds to the receptor, like that, we get the response. Make sense? Okay. Now, ligand protein. Yeah? Specificity, affinity, competition, saturation, all that applies. Okay. What's the response? Depends. Depends on what this is. We know from is this a signal transduction? Sure. Is this neurotransmitter coming inside the cell or is it binding to a cell surface receptor? Cell surface receptor. So we have to transduce the signal. Now, we saw that there are, maybe we didn't see, but we mentioned that there are three really common ways that a hydrophilic signal like that could work. It could be bound to an ion channel, it could be linked to an enzyme, or it could be a GPCR coupled receptor, right? G protein coupled receptor, which would trigger a second messenger response. In general, we break them down into two categories here. If the receptor is ionotropic, tropism means to give rise to, right, or to affect. What's it affecting? Ions. These are going to be linked to ion channels. Metabotropic receptors are going to be those that work through a signal transduction pathway involving a regulatory protein, a G protein. So ionotropic receptors are linked to just an ion channel. And it binds, what does the channel do? Opens or closes, and you get ion flux. That would be very rapid, right? Channel opens, channel closes, rapid responses, ionotropic. Metabotropic receptors, on the other hand, are these ones that involve a cascade. That's the metabolism. A happens, and B happens, and C happens. And these are G protein coupled receptors, so these are GPCR. And they're going to be a slower response simply because there's more steps that have to happen, right? And it depends on what they do. Sometimes they open and close ion channels, sometimes they trigger gene expression and cell division, depends on what the scenario is. So these receptors, these postsynaptic cell receptors, will fall into one of these two categories. Ionotropic if it opens or closes an ion channel, metabotropic if it's a G protein coupled receptor. It's just a way to divide it. Now, could the same neurotransmitter bind to an ionotropic receptor and a metabotropic receptor under different conditions? Absolutely. Why not, right? Certainly they could. It just depends on what the postsynaptic cell has. The presynaptic cell releases a neurotransmitter. A receptor will be one of these two categories, and it will trigger a response. And the same, a neurotransmitter like acetylcholine can have a variety of different effects depending upon what it's being used for, where in the body is it being used. So it really just kind of depends. Now, neurotransmitters are paracrines. Right? They're released by one cell, they move through interstitial fluid, and the receptors are on adjacent cells. So specifically, since it's a neurotransmitter that's a paracrine, it would be called a neurocrine, right? But it is not a hormone. There are neurohormones, and those do not work this way, right? They're not released into a synapse. They're released and absorbed by the blood. Now, they're grouped based upon their chemistry. And again,